I would like to announce the next speaker is Adam Butterworth from University of Cambridge. I think it's the Department of Public Health. And Adam is going to be talking about integrating genomics and multi-omics at population scale for disease and size.
Great, thanks very much. So thanks for the invitation. As the slide says, we work in the public health department, so we're mainly interested in populations and largely healthy people rather than the patient in front of me. I'm a genetic epidemiologist by training, so I'm interested in how we use genetics and other layers of what we call multi-omics data to understand about disease. And I'm not a mathematician, there's not a lot of algebra in these slides, but what I've tried to do is throw in some challenges because I can see we use very simple mathematical or statistical tools, but I can see lots of areas where actually bright people with creative ideas could come along and improve on the way we do things. So I'll flag those up as we go along. So broadly, as I say, we're doing very simple things. We're doing things like we have a particular interest in cardiovascular disease and hence interested to hear about the center's focus in that area. But I'm interested in things like how can we work out which of the 3 billion base pairs in our genomes, which ones seem to predispose people to having heart attacks or not. And we can do this very simply by collecting groups of people who do and don't have heart attacks and then looking at doing association testing. So using kind of simple logistic regression models with a few covariates thrown in to say which of these seem to be most statistically associated with risk of disease. Or we can do that in the frame of a quantitative trait. So we now measure lots and lots of parameters in the blood. You heard earlier about the full blood count data in our interval study. We've measured lots and lots of different things like proteins and metabolites and gene levels. So we can take all of these quantitative traits and say, well, let's look and see for each of the genotypes that we have measured, each of these different genetic variants, which ones seem to be most associated with levels of these things. So here's an example of that. This is looking at heart disease from recent work we've done. And so, of course, instead of looking at one variant at a time, we tend to look at millions of variants. So maybe this is where some of the sort of scale problem comes in. We've heard about things like imaging data, where we have lots and lots of data points. Nowadays, we're talking about things like, in this project, I think we had about 11 million different genetic variants that we were testing, but we've now, as a community, discovered probably 500 million different genetic variations. So there's lots and lots of tests that we can do. And so this plot is kind of showing you the results of lots of those logistic regressions. So each circle here is a particular genetic variant in one particular region of the genome on chromosome 11. And on the y-axis here, we're just plotting the negative log 10 of the p-value, so a measure of the statistical significance. And so you can see in the middle, there's a nice strong signal here that we have. So this reaches that the horizontal line is our kind of multiple testing correction that we conventionally use when we're testing the whole genome at a time. You can see there's a nice strong signal in the middle. There's lots of the points are colored by their correlation. So we have a correlation structure problem as well here. You see that kind of that peak in the middle of all those clustered variants. Those are all highly correlated variants. We know there's some sort of signal there, but we don't know which particular genetic variant is causing the risk of disease. We just know that one of those is involved. And you can also see over to the left, there's probably another separate signal colored in the dark blue. So uncorrelated with those lead variants, there's we're starting to get to the kind of problem where any region of the genome you look at, there's probably multiple signals with multiple different diseases and traits. So we're starting to get it's great. There's so much signal coming through the noise. You can see all the noise down at the bottom, many, many null variants. We're starting to get to the sample sizes where you can disentangle those, but we're starting to get the opposite problem, which is now anywhere you look, there's lots and lots of signals, and that's causing us some problems as well. So this is if you expand out and look across the whole genome. This is a project we've done recently that's coming out soon, which is about 1 million people from around the world. About 200,000 of them had some sort of heart disease, and the remaining 800,000 were disease-free. And you can see many, many peaks on different chromosomes here. The black and gray ones are ones that we knew about previously, and the green ones are the ones that are now reaching our multiple testing correction. So out of the 11 million or so we looked at, there's about nearly 300 that are kind of independent signals. And if one's prepared to be a bit more lenient, we know the rest of those ones that are below our threshold aren't null. There's some real signal in there. So maybe there's something like nearly 1,000 signals if you took a 1% false discovery rate. So, what we've learned in the last 20 years is that diseases like heart disease are hugely what we call polygenic. There's bits of the there's across the whole genome, there's statistical associations. The other thing we've learned is that actually, because of that, most of these signals individually are very, very small. So here we're plotting each different variant that we found there. You've got the blue and red ones are the kind of ones we found, and the yellow ones, the robust ones. The gray ones are the ones at the 1% false discovery rate. And if you look at the y-axis here, this is plotting for each variant what the odds ratio of that variant is. So how much of an effect on risk of disease does it have? And you can see that they're largely now less than 5%. So carrying or not carrying that individual variant really affects your individual risk very little. So it doesn't tell me, if I look at one of those variants, it doesn't really tell me very much about whether I'm at higher risk or lower risk of heart disease. But Jessica told you earlier about predicting or screening people and trying to predict heart disease using traditional risk factors like lipid levels and blood pressure and BMI and things. You may be familiar with polygenic risk scores. This is the idea that for many diseases now, we can capture this genome-wide genetic component. We can say an individual variant is often not very useful, but because there's hundreds or thousands or perhaps tens of thousands of variants that all have very small effects, we can aggregate these. And again, the maths here isn't complicated. These are weighted sums. We're taking which genotype you carry at the first variant and what its effect size is, and we're kind of multiplying that through. What this plot's trying to show you from our recent paper is what happens when you scale up. So the 2015 PRS, the red dots here was from an earlier study with about 60,000 cases of heart disease. The new one, and that was the best performing score. We tried about 800 different combinations, which probably was very carbon expensive. But the best performing scores here from the 2015 data was a 1.4 million variant score. From 2022, we needed slightly more variants. And what you can see, we're using just the hazard ratio here, but the same applies if you want to use a C-index or other measures of kind of predictive accuracy, is that we get some incremental improvement in predicting future heart disease risk. It's not huge gains. Whether on the left here is people who have never had a heart attack, who go on to have their first event. On the right hand side is people who've had pre-existing heart disease and their risk of future events. But there's some improvement by having a much bigger starting data set. It allows you to separate out that signal and noise somewhat better and get better prediction. But I think there's much better things we could be doing here. We're using relatively simple methods. So first of all, there are now lots of different ways. How do you distill those 11 million variants that all have some kind of p-value? There's lots of null ones, there's lots of strong ones, but all those ones in the middle, how do you decide which goes into your score? How do you deal with the correlation? Our approach was to run several hundred with different kind of parameters and see which one came out top. But I think there's definitely better ways one could do this. So people haven't looked, for example, at how you combine different methods. Can you learn from the strengths of different approaches? Currently, we're treating all of these variants independently. So we kind of tend to use methods that remove the correlation structure and say, actually, we don't want these correlated variants. We don't want to include the same bit of information twice. But actually, how could we do better? Can we allow, for example, also think about interaction effects? Currently, we're treating everything one by one. We've also done some work, Mike Inouye, who Loic works with, has done some work suggesting that you could do better by not just taking our genetic analysis of heart disease and using that as the basis for the predictive tool, but what about if you took all those risk factors? If you add in a prediction for your cholesterol levels, for your blood pressure, for your BMI, you can slightly start to improve these tools. And I was very interested to hear Rachel talk about biases in machine learning algorithms. I think we need to be much more cognizant of the fact we have biases in all kinds of clinical algorithms. So polygenic risk scores are rapidly heading towards the clinic, and there are trials of them going on in different areas right now. But of those million participants I showed you that we use for the heart disease study, probably 90+% of those are people of white European ancestry. And so these polygenic risk scores, we're testing, we're deriving them, we're training them, we're testing them in white people. And of course, when you take that score and then try and transplant it to other ethnic groups, it performs less well. So we could sit around and wait for a decade or so to find 1 million people of each different ancestry group. But ultimately, I think we're going to have to shortcut this. It's no good giving clinicians scores that work well in white people but don't work so well in other ethnic groups, because it's just going to exacerbate health inequalities. So again, I think statistical methods, and there's an emerging field of work which is trying to work out how do we overcome the poorer performance or the features of the training set to make better scores that will be more universally applicable. So I think there's lots we can do in the prediction area. I'm going to focus more now on, most of my interest isn't in the prediction side, but it's actually in how do we go from the statistical side. You know, it's no good if I want to prevent or treat cardiovascular disease if I've got lots of statistical values. I can give you a nice table of p-values, but it's not going to help anyone. So how do we go from understanding that there's some statistical association here to working out what's the biology, what's the causal factor that's going on here? You can see at the bottom of this plot, there's quite a lot of different genes that are drawn. These are these acronyms with the arrows. The problem is that we don't know for any particular variant on this plot. We know there's a signal there. We have robust statistical evidence, but we don't know how to work out which of these genes is the one that's the causal actor in this region, or rather, is there in fact more than one gene? This is something we did in our recent paper to try and address that, because you know, in previous papers, we tended to be a bit hand wavy. You know, you'd have a PhD student who'd go and find all the papers they could on the different genes, or look in some databases, and you'd kind of write a paragraph about which one seemed most compelling. We tried to do this in a more systematic, automated way now. So we came up with an approach where we had 8 different predictors. We took a training set of 30 kind of positive control genes, ones that we were pretty sure kind of gold standard set of ones where we knew the answer. And we had some good ideas about what predictors might be. So for example, the nearest gene to the top most associated SNP is, depending which paper you read, something like 2/3, 3/4 of the time is likely to be correct. We know that if there's a rare gene that's nearby that carries a really rare mutation that leads to a rare form of cardiovascular disease, that's more likely to predict the right gene. And so you can see in the columns here of the matrix, we kind of had these 8 different approaches, and we kind of scored each region. We took each gene in each region and scored them according to where there was evidence. And we debated a lot about what's the best way to then integrate that information. You can kind of come up, say each predictor on its own is useful, and you know, we can give a yes, no, a red or a white for each, but how do we integrate them? For the sake of simplicity, we actually just summed them. So we just said, you see on the right, the kind of distribution. So there were 3 genes for which 7 of the 8 predictors lit up on that gene. So that was pretty high evidence. And again, you can then kind of debate where you think is sufficient, how many predictors you need. But we just took a kind of counting approach, and you know, happily, it works well for many of our positive control genes, which is possibly a biased subset. You know, some of these predictors probably are what's caused us to think those are reliable genes, but hopefully this is going to help us turn those 300 signals into some useful biology. Again, I think there's lots of better ways we could do this. We kind of debated whether there was better statistical models. I think the obvious thing to do would be think about how you weight the different predictors differently. So we just kind of summed them up, but you know, is nearest gene as valuable as having a rare disease gene or having any of these others? I think one could think about that. One of the ones that I should have put on this slide, but I didn't, is we tend to think about one-size-fits-all approaches. We tend to say, you know, for this region, score all the genes, which one's top for this region. But actually, we know that there are some where the reason we know that's the causal gene is because of one very specific piece of evidence. And actually having exclusions and saying, well, you know, if you have this predictor, this one's probably less likely, for example, or where does the model break down? I think the model worked well for, I think it correctly got 28 of our 30 gold standard genes. Why doesn't it work for the other 2? How can we tweak this to make it better? So I think there are different ways we could do this. Again, coming back to what I said earlier, integrating information from related traits. We've worked on genetic studies of lipids, for example, where we can actually go deep into the biology and what's known about metabolism and biochemistry, and we can be pretty certain what the key gene is. So one could then say, well, you know, if a gene affects cholesterol levels, it's likely to be the same gene, the same region here that's affecting heart disease. So we could probably have a pull through of information in some way as well. And of course, people tend to, you know, do one paper at a time. They publish the genes they think are most relevant for this one, but how do we integrate or synthesize across the literature here? There's a group called Open Target, which is a public-private partnership with some pharma companies down at the EVI and the Sanger Institute. They've thought about this in a very global way, so they're kind of phenotype agnostic, but they've built this kind of nice machine learning model that they train, again, using kind of gold and silver standard sets. that works well, kind of generally across phenotypes, but it doesn't necessarily isn't the best for any particular phenotype. So we had some examples here in our paper where we said actually, because of the evidence we're bringing in that's maybe more cardiovascular specific, or because we weighted differently, we think we've got the right answer. So are there ways that we can improve on the model? We can take the benefits of their statistical approach, albeit we have very limited training sets here when we stick on one phenotype. So how could we improve in that space? So I think this is an area, it's been, we pre-printed this about a year ago now, and there's now several other preprints of people who've used, almost exactly the same figures and the same approach. So it's good that people are kind of being more systematic about it. But I also feel like we used a very basic model, and that actually we need to kind of rapidly improve on this so that people aren't just starting with our initial starting points, but are actually doing something a bit more valuable. So going beyond just learning about which bits of DNA affect your risk of disease, one thing my group's been particularly interested in more recently is kind of filling in the gap between DNA and disease, as you see here. So we can now measure at scale in people various different biological domains, and we can capture a lot of information here. So for example, we can sequence almost all of the genes, at least in we've done this in people's blood cells. You can see the gene levels. You can measure several thousand proteins now that are floating around in people's blood. So the technology has really improved, not to the scale of genetics, where we can capture almost everything with really high fidelity, but to the scale of we can find a lot of meaningful information. And I'll give you a few examples of how we're doing this. There's two main things we can do once we link the DNA with these molecular levels. One is called co-localization. We say, if we look across the whole genome, I gave you those 300 heart disease signals. What other signals for different domains of molecular traits line up? And by co-localization, we have to kind of account for that correlation structure and say, are these two signals really the same? And we can put that in a formal statistical framework. And the other is a concept called Mendelian randomization, which is trying to infer causality of risk factors, which I'll explain in a minute. And somebody mentioned earlier the interval cohort and the blood cell trait measures we have, but we've collected 50,000. These are blood donors, so they're largely healthy people, but we think they're still informative for learning about disease. Some of them, of course, experience disease events, and they're all linked to some of the hospital records and other NHS systems that you heard about during the course of the day. But we've particularly focused on things we can measure in their blood samples. So as you see, kind of several different protein assays and metabolite assays, and thousands of different molecular traits, basically. What can we do with this? This is one of our first examples. We used a particular platform called SomaLogic. This is a company out of Boulder, Colorado. It was one of the first to work out how you could try and measure 3,000 different proteins in the blood at scale with some degree of reliability. And what we did is we took the whole genome and we then correlated. We did all these association tests. As I said, the maths isn't complicated. We sometimes get mathematical interns or PhD students who come to the group who get very disappointed when I write down the statistical model we're going to use and say, the model's not complicated, but we're going to do it millions of times. So this is 10 million genetic variants on the x-axis here against these 3,500 proteins. And so each point is basically a statistical association between the genome and the protein level. And so what you find here, the red diagonal line is showing you what we call cis variants. These are genetic variants that lie near the gene that codes for the protein. So often you look at the whole genome and the only place there's a signal is near that gene that makes that protein, which makes good biological sense. But perhaps more interesting for understanding about biology is the blue ones, the ones that lie somewhere else in the genome. Why does this other bit of the genome seem to affect this region of this levels of this particular protein? And sometimes they make a lot of sense. It might be a ligand receptor pair, they might bind to each other or something. But you can also see there's quite a degree of pleiotropy here. So if you look at the, you might be able to pick out some blue columns that we labeled the names for at the top. There are some regions of the genome that seem to have hugely pleiotropic effects. So one of them on chromosome 9 is the ABO blood group that seems to just distort all kinds of molecular factors. There's the ApoE region, which is related to kind of lipid metabolism, seems to have pretty effects on heart disease and Alzheimer's and all kinds of other traits and diseases. And so this is kind of interesting. It was like one of the first studies that really showed which bits of the genome control levels of which kinds of proteins. But we wanted to then do something useful with that. So if we go back to my region related to heart disease from our recent study, and I gave you the problem we have earlier of all these different genes in the region, how do we know which one? Well, for this one, we can see that if we look at the protein levels, there's a very nice signal in the same region that overlaps very nicely. Those same set of genetic variants seem to also associate with levels of this plasma protein called SWAP70. You can see the gene down below it there. And so it seems very likely that this genetic signal for heart disease is acting via levels of this protein SWAP70. It's given us an anchor to go and explore the biology, to go and design wet lab experimental studies to say, well, how does this influence risk of heart disease? I've alluded to this already, but of course, one of the things we want to do is we can get very distorted or confounded by the correlation structure here. So, if I just quite a lot of the time, if I just take the top variant here for heart disease, and if I just look it up in lots of the published literature, I will be bound to find lots of small p-values. But what I don't know when I just look at that variant, I don't know if I'm picking up the top of a peak of association, or I'm just picking up one of these, perhaps the tail, if you like, of a big peak. So what we use is these co-localization methods where essentially we're comparing two different traits and saying, does the signal look the same? Or we could put them on a biplot as in the bottom left here and say, essentially we're testing the correlation here. What's the correlation of the variance of the association statistics between trait 1 and trait 2. And there are various methods now to do that, usually using a kind of Bayesian framework and putting some priors on them and kind of comparing different hypotheses that the two traits do share a signal, they don't share a signal, etc. I alluded to the problem before that now what we're finding is that you don't tend to just get one signal in a region. So of course that correlation, that testing really breaks down. If you start to introduce multiple signals in the region, that nice kind of y equals x is going to get distorted and your co-localization testing model goes all over the place. And so we've kind of so far found fudges, if you like. I think I can say that I was involved in designing this in this paper, where we kind of said, well, this is a problem. So what we're going to have to do is we're going to have to condition out all the independent signals in the region and just test them one at a time. So in this instance, we were trying to do lots of diseases against lots of proteins. And we said, well, to make it feasible, we'll just try and, if you've got at the top, you've maybe got a region with 2 different protein signals. And at the bottom, you've got a disease with 2 different disease signals. We'll just kind of condition them out and then do pairwise testing of all the combinations. And then that will tell you, which ones are the same, which ones are different. And it was useful, and it helped us find some more that we wouldn't have found previously, but it's less than ideal, I would say. And here's an example of where it kind of goes wrong. So that same SWAP70 region I've shown you, I've told you it's associated with the heart disease. I've told you it's associated, and it seems to be the same signal with plasma SWAP70 protein levels. There's also a signal in the region for blood pressure. And again, that would make sense. That might explain, maybe the alterations in SWAP70 protein are changing your blood pressure, which is changing the risk of heart disease. But when you put this into a co-localization model, it tells you it's not really the same signal. And that's because on the right-hand side here, you've got this kind of dominating, this even stronger independent signal. And so if you just take that whole region and put it into your co-localization testing, it tells you now these are separate signals. But actually, once you condition out that second signal, which is the approach we were using, it leaves the nice red signal in the middle. It tells you it is the same one. So again, I think there's great room for kind of methods development here, particularly around methods that can account for having multiple signals in the region. It's only going to become more of a problem as the sample sizes grow and we find more and more true signals. How do you work out in complicated regions which signals are truly the same and which ones are just confounding because of the correlation structure? There is now one method that can handle multiple traits, but increasingly, we're finding signals for heart disease that then also have a gene signal, a metabolite signal, a protein signal. How do we test these jointly? And we've alluded already to this kind of ethnic bias or ethnic differences. One of the challenges is the correlation structure is relatively stable within an ethnic group, but once you start to go across different groups around the world, that correlation structure differs. And that can be useful because we can compare and we can help narrow down signals. But when we're thinking here about co-localization testing and incorporating, accounting for that correlation, how do we deal with the fact where we might have data on the disease from one ancestry group and the trait we're interested in from another ancestry group, or some different proportions of different ancestries, for example. So again, I think there's great scope for improving in how we try and answer what's seems like a relatively simple question. Is this signal the same across different outcomes? The other approach I mentioned that we like to use is called Mendelian randomization, and that's the idea that ultimately you might want to do a big randomized trial. You might want to randomize people to a drug that lowers levels of this risk factor and a placebo, and then you're going to compare the event rates at the end of the day. And that's a very long and expensive process. What we can try and do is take advantage of genetic inheritance and say, actually, people randomly inherit these alleles from their parents, and therefore through a population, it's effectively randomizing people. So if you can pick a genetic variant that's associated, say, with levels of a risk factor or a protein, and compare groups of people with different genotypes, then you're effectively saying we've randomized people naturally rather than with a drug. People should have different protein levels, and therefore can we compare the outcomes? So it's trying to kind of mimic a trial, but in a natural way. And here's an applied example of that. We took the somologic proteomic data I showed you, and we said, this is one protein called matrix metalloproteinase 12, or MMP12. And we were lucky because we found lots of different genetic variants in the region near the gene. So they seem to be very specific to that protein. And so on the x-axis here is each variant's effect on the levels of the protein. And on the y-axis, we were then plotting their risk of coronary heart disease. So this is taking data from different data sets, from the interval for the proteins, and then a global meta-analysis for the heart disease. But you can see there's a clear consistency here. So if you happen to inherit a genetic variant that gives you high levels of MMP12, you seem to have lower risk of heart disease. And this was quite a surprise to us when my PhD student first kind of showed me this plot, because the literature that had been previously that had looked observationally showed that people who have higher levels of MMP12 have higher risk of future events. And so initially we were kind of checking we'd got things the right way around, the right genotypes, the right alleles, the right beta coefficients, etc. But actually the latest literature seems to suggest this is probably because it's a response biomarker. It's a protective factor that's being released by your cells in response to the vascular damage that's predisposing you. So if you have high risk, this is a kind of warning biomarker, which is why the higher your levels, the higher your risk of a disease, but that's because your body is trying to protect itself. It's trying to repair that damage. And so one can start to turn observational insights into what we think might be causal insights, and can start to use this then to think about how we might target, how we might develop treatments for disease. And rather than this is a one at a time approach, looking at one protein, but we're now kind of scaling that to genome-wide or proteome-wide approaches. So this is a paper we did last year in response to the COVID pandemic, where we said, well, let's try and create as many different kind of randomized trials as we can, take as many genes and proteins as we can find good genetic variants for. And then let's use the data that's been rapidly thrown together by the global genetics community on risk of COVID outcomes. And then let's try and do all these different Mendelian randomizations. So this time each dot is a gene or a protein. And you can see some of the regions of the genome that started to light up in terms of signals. Reassuringly, on the right-hand end there, you see ACE2 on chromosome X. That's the protein that acts as the receptor for the SARS-CoV-2 virus. So that gave us some confidence our model was working. When that came up, that was already known to be involved. But some of the other signals were then potentially interesting, particularly where there were drugs that could be potentially trialed or repurposed. So, for example, IFNAR2, which is interferon response related gene, there were drugs that were being considered for trials for that. So one can start to get relatively rapidly actionable insights by integrating the genomic data, the proteomic or transcriptomic data, and outcomes data. So I didn't put a slide, I forgot to put a slide in about challenges for Mendelian randomization, but I think some of the same approaches that I've already talked about are challenging here, not least the fact that this only really works if you have a genetic variant that only affects the protein or the risk factor you're interested in. So if you really want to randomize assume that the two groups of people who carry the variant and don't carry the variant are really effectively randomized, there's no confounders that differ between them, you need a variant that really only affects the risk factor you're interested in. And so finding these tools and working out which ones, using that kind of co-localization strategy to say, this variant seems to have lots of small p-values with lots of different traits, but how specific is it? Is it very pleiotropic, in which case it's probably not that useful, or is it actually very specific to the thing I'm interested in? So that's one of the main challenges we face in these kind of approaches. So hopefully I've convinced you that some of the very large scale genetic studies we're doing are starting to tell us about links between not only statistical links between genetic variants, but also pathways, biological information, which are molecular risk factors for diseases. And people often get very hung up on the fact that these individual variants have very small effect sizes. One of the slides I didn't put in today is one of those 1.03 odds ratio variances in the HMGCR gene. The HMGCR protein is the target of statins, the most effective therapy we have for cardiovascular disease. So just because natural genetic variation has a very small effect, it doesn't mean that pharmaceutical approaches can't then come along with a much bigger effect on that pathway, completely knock down or knock out or upregulate a pathway and have a meaningful disease effect. So yeah, I spend a lot of time telling people small genetic effect sizes don't matter. As you've hopefully seen as well, prediction can also happen. We can overcome the small effect sizes by huge numbers of variants that are probably associated. And I think we'll do better with that over time. And I haven't even shown you the statistical models we're using because they really are very basic, but I think that means there's huge scope for improvements in these things we do. We're taking very simple methods, applying them at very large scale, large populations, large numbers of risk factors, large numbers of variants. We can look at large numbers of outcomes, but I think we can definitely therefore improve on. And the other thing I would say is that these are all almost all one domain at a time approaches. So I showed you a genetic analysis of proteins and one of transcripts, and we have, each of those ones I showed you in the interval cohort, we have a separate paper on. What we don't have yet is that fully integrated. People show you nice slides of how we're going to integrate all these domains of information. Of course, biology doesn't act in silos. It doesn't act in these individual domains. They're good starting points, but I think the next frontier is how do we start to integrate that information? So how do we look at the individuals who carry disease, who don't, and what's going on at a systematic multi-omic level. So of course, this is the work of many, many people in Cambridge and across the country and around the globe. So and it's funded by many different bodies as well. So I should acknowledge those. I'm happy to take any questions. Thank you.
Thank you. Very enlightening talk. And very heavily loaded with genetics. I've learned more than I did in my previous life. Any questions from the audience first?
Thank you, wonderful talk. So I was wondering when you're talking about the co-localization of the different traits. So does that mean the different traits can have shared genetic underpinnings? And have you tested any kind of the pathway individually or shared way?
Yeah, exactly. And that's one of the things we're often looking for. So sometimes it might be two diseases and we might be interested in saying, actually, this is interesting because this signal is with two maybe similar diseases, or even more interesting, two completely unrelated diseases. And that often gets us excited, why? I'm particularly thinking about, well, there might already be a drug for this particular protein in one disease, but it's never been considered in another disease. It could be so we get very excited about that, but perhaps more so about, if you think about each genetic variant, it has some very local function, right? It's doing something to the gene or the protein, okay? It's going to have some slightly more downstream function. It's going to have something that, some area of biology, metabolism or immune response, before you get to that end disease. So what we're trying to often do is piece together, work out which of those bits in that sparse matrix do overlap and which ones are really real signals. What we find nowadays, particularly say with gene expression, there are now data sets of maybe 30,000 people, and almost every genetic variant you look at in the genome has an association with at least one gene, often multiple genes. And so what we're starting to have to try and do is see, where are these peaks, which ones actually line up and are the same signal, and which ones are just noise parameters and are not really the same signal, but they're just correlated with a nearby signal. So it's starting to try and dissect those regions, because you can make very inappropriate inferences if you don't actually check for the same signal. And then what you can do, what's nice if you have them in the same data set. So for example, in interval, we can do this well with the molecular data, is you can say, well, we can start to do sort of network or mediation analysis. So, you know, if you condition on the gene transcript level, does that affect your association with the protein level? So, you know, you can start to do those things. And again, we're doing those in very simple kind of one at a time ways, but one can start to imagine much more kind of network models and much more complex models, multi-gene, multi-trait, multi-protein models. Ideally, you want the disease event in the lots of the same people as well, but probably an interval for now, we'll probably have to stick with the healthy people and the molecular layers, but you know, those data will come. Yeah.
The correlation between RNA and protein expression is often only partial. How do you think we can overcome the challenges this presents for multimodal integration?
Yeah, that's a good question. Every time I talk about the protein results, people always ask me how it compares to the RNA results. And that was one of the things we tried to quantify in that paper, because people are very interested in, you know, how often does a signal that affects gene expression affect protein levels and vice versa? And again, the answer was only about 40% or something like that. So one of the key limitations here is all the work we're doing here is in blood. So much of the time, maybe, you know, the protein level in blood isn't determined by gene expression levels in blood. It's, you know, gene expression in the liver that's then controlling protein secretion into the blood. So there's, I think one of the problems is that we don't have a complete picture yet. But there are lots of nice examples now where actually they are sparse, because it's, you know, there's a variant that only acts at the protein level. It doesn't act through gene expression. So you don't expect to see an expression QTL, but you do see one at the protein level. And actually, sometimes it's nice when they all line up and you can make some sense of it. But often we know that there would be variants that change gene expression that don't act on blood proteins, and variants that affect blood proteins. So I think there's sometimes interesting information to learn by absence of effect, as well as, you know, it's nice when they do all line up, but often the fact that they're only in one or the other tells you something useful about what's going on in biologically.
Thank you for this very interesting talk. When you talked about this pairwise conditional co-location method, do you think that there is a chance to relate it to sparse causal graphs or do calculus?
It's not a question I'm equipped to answer. I'm equipped to apply the tools, but I'm always willing to hear solutions to it. So yeah, we took an approach that we could kind of handle using the data we had. One of the challenges you often have here is you often have only the summary statistics, by which I mean for each variant you only have the beta and the standard error, so you don't have the individual level data. And that means we often have to use approaches where we have a list of variants and their summary statistics, and then we have to bring in a correlation matrix from something else that somewhere else we know. So we're often having to use approximate approaches rather than exact approaches, which is often a challenge and a limitation. But yeah, I think there's real room for innovation here, for somebody to kind of say, I've got my head around the problem here. I can see the data you've got, and here's how we could kind of think about it from a completely different way. Because of course, you know, people in statistical genetics build on each other's work, but actually I think there's room for kind of serious disruption here, if you like.
Thank you.
Any more questions? I have a question following from what you said about the current thinking is that the genetic effect size is very small comparatively to the factors. So basically, what is your take on, is it nature or nurture? And so is it how we behave? Is it the like lifestyle factors? So is it actually the genetics that determines the likelihood of cardiovascular disease?
Well, both. I alluded to the fact that these genetic predictors can be quite useful. I mean, sometimes we think the genetic, the polygenic risk score would be useful because, for example, you can measure it at any age and you only need to measure it once. It's fixed, so it has some utility in that. But we also have shown that it's useful. It still predicts disease even when you bring in traditional clinical risk factors. So for cardiovascular disease, you know, even if you have a model that includes, age, sex, obesity, lipids, blood pressure, the genetic effect still brings something. That's partly because it's capturing pathways that aren't already measured by the traditional risk factors. I think it's partly because there's measurement error in the individual risk factors that you're picking up. You know, your lifelong predisposition to high cholesterol isn't necessarily captured so well by a one-off measurement. So yeah, there's clearly both going on. What we haven't found yet as a field that well is people had this idea there were going to be lots of gene lifestyle interaction effects, and we did grants and projects to do with that. They've been very difficult to find. I think we're now getting a good handle on the main effects, but actually probably because those main effects are so small, you know, the chance to find interaction effects in those ones is very difficult, because you're talking about a variant that might be, you know, odds ratio 1.02 in this scenario or 1.06 in this scenario. So those will be challenging to find. So yeah, definitely both. There's genetic and lifestyle contribution, but
Also like
yeah.
smaller population sizes you're looking at.
Yeah, exactly. And you know, we are now getting the sample sizes. When people are talking about now analyzing millions of people in genetic studies, you can start to see really robust evidence of these interaction effects. But as always, that comes after the discovery of the main effects, which is less power hungry. Yeah.
Thank you very much.
Thank you.
Yes, I think what remains for John and myself to say is thank you. Thanks a lot for a great day. Thanks to all the speakers.